Right. Biotechnology, here we go. All right, so you guys have your exam coming up. The exam is going to cover chapters 8, 9, and 10. You have a review sheet. Be sure that you use the review sheet. The review sheet is very valuable for this exam. If you go through each of those 40 items and define it, for the most part, you are going to be A+ for your exam. Right? So also I made a little modification to the syllabus, guys. I took off a couple chapters at the end here. They're really not too relevant. So we're actually going to just do one more chapter, and that will be the conclusion of this course. You guys have done very well. I've started grading your papers. They're excellent. For the most part, you guys have done really solid work here on that. Excellent effort. Really appreciate that. All right, so chapter 10, Aquatic Biotechnology. So this is a pretty cool, very rapidly growing area of biotechnology. And it takes, the tools that we've learned about in genetic engineering, molecular biology, and other biotechnology techniques, and it brings it now to the aquatic environment. So we're going to look at how aquatic organisms are being used to increase the global food supply, develop medicines, create industrial products, clean up our environment. So look at what we're going to cover here in this chapter. We're going to look at an intro to aquatic biotech. We're going to move into aquaculture and how aquaculture enhances food production. We're going to look at genetic technologies applied to aquatic organisms. We're going to look at medical applications, non-medical applications, and then finally, we're going to look at how aquatic biotechnology contributes to environmental solutions. All right, so let's get started here with, so let's here. Let's start with aquaculture. All right, so aquaculture is basically the controlled cultivation of aquatic organisms. These are things like fish, shellfish, including aquatic plants, and using all this stuff for food, recreation, or getting it commercially, selling it. So when practiced in marine environments, it's called mariculture. And you can see here in this figure, aquaculture can involve like some really large scale operations. This is a net full of farm raised catfish ready for market. These industries are expanding rapidly as we try to meet global food demands, and it relieves pressure on wild fish stocks. All right, so understand the economics of aquaculture are important, right? So according to the Food and Agriculture Organization, the demand for aquatic products is expected to rise by 70% over the next few decades. But nearly all fish stocks are already fully exploited, and around 30% are overexploited or recovering. So more than half of the fish consumed globally today now comes from aquaculture. This industry really is no longer supplemental. It's completely essential. So aquaculture, though, it isn't just about like dinner. It provides bait fish for fisheries and small fish like anchovies and sardines. These are harvested for fish meal and oil. These are essential ingredients in livestock and aquaculture feeds. But really, a very big array of aquatic species have been successfully cultured, demonstrating the versatility and potential of modern aquaculture today. So aquaculture practices vary significantly. Factors like the species being raised, the life cycle of it, environmental needs, and the time required for them to reach market, all these influence farming methods. whether it's shrimp in tropical ponds, trout in freshwater ways. Each system, has to be tailored to the biology of the species. So to ensure optimal results, breeder stock is carefully selected based on desirable traits like rapid growth, disease resistance, meat quality, and even color, right? Coloration. So think about it. This form of selective breeding really helps farmers improve productivity and consistency, ensuring that each generation is better suited for aquaculture. Fish farming, so it often begins with fertilized eggs in small tanks. Once hatched, the young fish, these are called fry, are transferred to larger tanks with flowing water to stimulate the natural environment. Culturing techniques are constantly being improved to increase yields and lower costs, making the industry more sustainable and profitable. So here, this figure here gives an overview of fish farming systems, from hatcheries to grow-out tanks and feed management. Every component is carefully managed to maximize output and minimize environmental impact and disease risks. So egg and sperm are harvested. Their fertilization is allowed in a controlled tank, and these fry or fingerlings, the fry are then transferred to like small pools or ponds and allowed to develop and grow. And then those are released back into the waterways. All right. So things like, innovations like polyculture have really reshaped fish farming. Polyculture involves growing different species in the same environment, species that complement each other in nutrient use and feeding habits. So this strategy really helps optimize water use and improve efficiency. Hydroponic systems go even a step further from that. They use fish waste to nourish plants, creating like a symbiotic ecosystem of fish and vegetables. So here you've got a polyculture system where tilapia and lettuce are actually grown together. And the lettuce helps control the nutrient levels in the water for the fish, while the fish provides nutrients for the lettuce. They're helping each other. Obviously you got a fish in a pond and it's just in there pooping. Eventually it's going to become poisonous, but the water's going to become poisonous from the ammonia, but the lettuce just soaks that up. So this is a very sustainable, space efficient, and a potential solution for like urban and resource limited areas. They got a really good demonstration of this at Epcot. I forget where in Epcot it is. I went there a few weeks ago and I don't remember, but it's pretty cool. They have, they actually grow, Epcot grows a lot of their own produce for the entire park. Okay. Anyway, so biotechnologists are also working on improving aquaculture species through what's called selective breeding, right? So here you kind of pull out desirable traits and promote the breeding of those traits to induce like rapid growth, improved taste, higher fat content, better fillet yield. So you could use things like ultrasound to help estimate these characteristics and without harming the fish. And they have these large ultrasonic machines that can analyze many fish at a time. It's not like you're going in there with and like analyzing one fish at a time. It's very automated processes that do these. So, all right, let's look at now genetic technologies. So, we've talked about a lot of these things in this course. So by studying gene expression and regulation in aquatic organisms, scientists are trying to improve traits like growth, reproduction, and resilience. There's also a lot of interest in understanding the immune systems of aquatic species. These immune systems differ significantly from terrestrial animals. So, gene discovery is really the cornerstone of marine biotechnology. As a good example, cloning the salmon growth hormone gene has enabled the creation of transgenic salmon. These grow much faster than their wild counterparts. Another amazing discovery, antifreeze proteins. So these lower the freezing point of bodily fluids in cold water fish. And these are being used to create cold-resistant fish and crops. Cool stuff. The all-famous GFP, or green fluorescent protein, the green gene. This is a really remarkable example. So this is found in the jellyfish, Achiura victoria, and this gene causes bioluminescence. So today we use GFP and this myriad of other fluorescent proteins that have been extracted from corals and things like that. And we use them as what are called reporter genes. So we use it to give us information about another gene. We'll show you how that's done here in the next slide. So basically, you take the fluorescent gene out of the jellyfish or coral, whatever. And these corals have all different colored proteins. You got everything here. You got blues, reds, yellows. If you ever go to like a saltwater fish store and you see all the different colored corals that they have in the tanks, those are all fluorescent proteins. Anyway, so you take the gene out from the jellyfish, and then you take the gene you want to study, and you put them side by side. You clone them side by side in a reporter plasmid. And you take that plasmid, you transfect it into cells and grow up the cells. And when this gets transcribed and translated by the cellular machinery that you're transfecting it into, it gets combined into a single protein. So now we have a protein that has the features of GFP, which means it glows, and the features of whatever gene we're studying. So here you can see, well, no pun intended, but you can see where the protein is in the cells. So excellent resource in biotech and molecular biology, cell biology. All right, so also sequencing the genomes of marine pathogens. This is a very important area of active research. For example, the genome of the P. salmonis, right? This is a bacteria that infects salmon, and it's been deciphered to help control the disease. We'd use PCR to detect DNA from oyster pathogens. This enables early detection and prevention of outbreaks. So here's some examples of PCR reactions that have been run out on a gel, looking for the different pathogens that infect oysters. All right, genetically modified aquatic species include transgenic and triploids. We're going to talk transgenics and triploids. We're going to talk about triploids here in a little bit, what that means. So all in all, though, these genetic modifications aim to improve growth, health, and environmental containment too. I'll explain that a little bit. So, but really until recently, no transgenic fish were approved for human consumption. But that is changing rapidly as the FDA further evaluates the safety and the ethics of that. So a little bit about ploidy, right? Let's talk about polyploidy. So if you remember back from our earlier chapters, most animals are diploid, right? You get one set of chromosomes from mom, one set of chromosomes from dad. Polyploid organisms, such as triploid fish, have 3 sets of chromosomes. All right, they have an extra chromosomal set. And these fish grow 30 to 50% faster, and they are sterile, so they can't reproduce in the environment. So we don't run the risk of them, mating in the oceans, and now we have an ocean full of mutant fish. This figure kind of represents ploidy. This is typical diploid here, right? And then we move up. We've got triploid and even tetraploid. There are species of plants that have polyploidy that's like up in the 25N, like multiple copies. And this is not an unnatural phenomenon. I mean, most tumor cells have polyploidy in them. All right, so how do you produce a triploid? Well, so you basically capture the egg prematurely or not the egg, but the gamete, prematurely during meiosis, right? So, during meiosis 1, chromosomes fail to segregate properly, right? So this is a rare event, but it does happen. So you'll have a gamete with no chromosomes and a gamete that's 2N, and you basically take that gamete and you fertilize it with 1N or 2N gamete, and then you end up with polyploid. All right. You can also do multiple fertilization, which is a little even more unnatural because you're actually subjecting eggs to multiple haploid cells. Usually you have to do that through microinjection or something like that. All right, so medical applications of aquatic biotech. So marine organisms, if you think about it, they are really a gold mine for drug discovery. We've talked about bioprospecting, the search of medically valuable compounds. Well, this is active in the aquatic systems as well. But it has led to many discoveries. The book gives good examples like salmon-derived calcitonin. Salmon-derived calcitonin treats osteoporosis. Adhesives from muscle-derived fibers have been used in adhesives that you can use in natural situations like bone repair. So, the oceans are really, they have a lot of tremendous biomedical potential. Right? Here's those byssal fibers that mussels produce, and they are useful in bone repair. So many invertebrates have yielded compounds with anti-inflammatory, analgesic, and anti-cancer properties. So a good success. So this prealt derived from cone snail venom treats chronic pain. Another one, Yondalis, is extracted from sea squirts. It fights soft tissue cancers. It's been in trials for other cancers as well. All right, got a lot of stuff. I don't really have any of these on the exam. I think, double check that real quick. Come back to the slide. Do I have that on the exam? Yeah, I got. So, homogenase, no. No, I don't really have any on the exam. Not on this list. Okay. Okay, another, the fugu fish. You guys probably heard of the fugu fish. So it's a Japanese puffer fish. It produces tetrodotoxin. This is one of the most potent nerve poisons on the planet. It's like 10,000 times more lethal than cyanide. Interestingly enough, this toxin and the fish is like a delicacy in Japan. Only when prepared correctly. If it's not prepared right, the chef doesn't know what he's doing. It's right to the emergency room or graveyard you go. But this is a really important tool in research for looking at nerve function and developing anesthetics, cancer treatments, Very big, important protein. Here's a little pufferfish. They look pretty happy though. Pufferfish, when they're scared, they puff up, and those guys don't look too puffy. All right, so there are other things as well. So non-medical products, aquatic organisms provide valuable non-medical products to enzymes like DNA polymerases, salt-resistant proteases are used in research and industry. Carrageenan, carrageenan, that is on your, what is carrageenan? That is on your exam, is derived from red algae. That's a common food additive, food preservative. Marine biomasses such as algae and plankton, these can be like tapped into for energy and carbon capture, offering green solutions to modern problems here. These enzymes you guys should be familiar with. We've talked about them in previous chapters. All right. And here's information on the aquatic biomass that I was talking about, algae and plankton. And you can use it for carbon capture and energy production. All right, let's talk about environmental applications. Oceans have been used as dumping grounds, but aquatic biotech can help reverse the damage. Anti-fouling agents prevent biofilms. Biofilms are layers of organisms like barnacles and algae that stick to surfaces, and they can cost billions of dollars in maintenance. All those ships, they need to be scraped. And there are certain marine species that naturally resist biofouling. These can offer like a template for an eco-friendly type of coating. All right, so this is showing how marine organisms have evolved natural mechanisms to deter biofouling. And studying and mimicking these adaptations can lead to new, less toxic anti-fouling products that protect marine infrastructure and preserve our ecosystem. So that concludes our lecture for this chapter. I wish you guys best of luck on your exam. So definitely use that review sheet that I gave you. All right. So the exam is very definition heavy. Okay. It's like probably about 70% of the questions are, what is this? All right. I've tried to make this one real straightforward for you guys. So look those terms up in the chapters, spend a little bit studying for them, and you are good to go with this exam. I'm expecting some real high grades on this. And then after that, we have one more left, guys, and the end is near. So congratulations on a job well done so far. And I will be in touch very soon with the next chapter.